If you've been watching this video series, there are probably some services that you thought were pretty amazing. And for me, generally, when I think of assembly, that is like the pinnacle of bioinformatics. You're taking millions of these little reads and using this pipeline, you're assembling them into contigs. Maybe 1,000, not good. Maybe 100, not bad. Maybe even just one. I mean, it's an amazing process. But as amazing as assembly is, the most powerful service that we have at BV-BRC, I believe, is genome annotation. That is taking those contigs and putting genes on them so that you can see what the function is, what your organism can do. Now, this is one of the most popular services that we have at BV-BRC. I mean, there have been over 700,000 jobs submitted to this particular service. And it's also a keystone service because you need to have an annotated genome to do a number of downstream applications in BV-BRC. And we'll go over those in a bit. But let's look at this service, and I want to show you how it works and what it can do. And in the whole series, we're going to be submitting an annotation job and looking at the results that we get from it. Although it's one service, there are really 4 pipelines. RAS-TK is used to annotate bacteria and archaea. Now, we will annotate any bacteria, any archaea that you throw at the service. We can do them all. Bacteriophages are annotated with a pipeline called Annotate. And like the bacteria and archaeal pipeline, we annotate all phage genomes. Now, the other viruses are a little bit different. First of all, we don't annotate at this point all the viruses. We concentrate on select human viruses. And the first pipeline that we run is called Vigor4. It will annotate select viruses. Now, if that doesn't work, if the contigs that you are submitting are not annotated by Vigor4, then the MAT peptide pipeline will kick in and we'll try to annotate it with that. But, if you've got some plant virus or something unknown, you may not get an annotation at this time in BV-BRC. So I wanted to show you what the RAS-TK pipeline looks like. There are a number of different components. It's pretty complicated. It's a sophisticated piece of bioinformatics software, if you want to get into technical terms. Or if you appreciate the arts, it's a beautiful picture of bioinformatics, linking these things together to give you this one polished product. So it starts with contigs that you submit to the service, and it calls tRNAs. Then it will look for repeat regions of different sorts and CRISPR elements before it goes to calling the protein coding genes, which are the CDSs. And then the whole thing is about annotating those proteins and mapping functions onto them. One of the things we do is look for certain areas that are associated with antimicrobial resistance. We also take every protein that is assigned in the genome and blast it against specialty databases. We annotate protein families. This is a pretty important function. We annotate cross-genus, crossing the genus barrier, and within genus protein families if you give us a taxon ID at the genus or species level. These protein families are important for other services, like the bacterial genome tree calls on those protein families to generate a phylogenetic tree. Also, the comparative systems service does the same thing in calling on the protein family. So it's an important thing. Once we have the protein families, we project the subsystems, we give it an MLST, we look for prophage genes, and then we wrap it all up into a beautiful packet of job results for you. The specialty genes databases that we look for, all the proteins are blasted against, There are specific categories or properties that you can see here. Antibiotic resistance is a big one. We also do drug targets, essential genes, human homologs, transporters, and virulence factors. Now, there are specific databases associated with each of those categories, and those are the sources of those. For example, CARD, the Comprehensive Antibiotic Resistance Database, is part of antibiotic resistance. TTD is part of transporters. Now, when you get the results, you'll be able to see how those things were mapped. It was either by BLAST-P, by BLAST, by the FBA flux balance analysis, which is part of the essential genes, a Kamer search, or literature. And if it is literature, there'll be a PubMed ID that you can actually link to. Bacteriophages are annotated using Phannotate, and you can see that it, like RAS-TK, has a lot of components and a lot of steps, and it starts with tRNA and CRISPR, much like RAS, and then the proteins are annotated and protein families. This is, among the viruses, pretty unique because at this time, we try to unite any annotated protein into a protein family. What this means is that some of the services that bacteria use, you can also use bacteriophages. You can't do that right now with the other viruses, but with the phages you can. So you could generate a tree using the bacterial genome tree because it's giving you these protein families. You could also use comparative systems. But speaking of trees, you could do the viral genome tree because it is a virus. And the viral genome tree service relies on differences in the nucleotide sequence, or the bacterial genome tree, which is looking at the protein families. So that's the bacteriophage pipeline. Vigor4, which is used to annotate some viruses, you can see it's simpler than the other two pipelines. And these are the viruses that are annotated using Vigor4. MAP peptide is even easier. And these are the viral families that are annotated using MAP peptide. Now it annotates 165 different species within these families. But I'll be honest with you, within these families, there are a lot more than 165 species, and I can't put it on a slide because it would just wrap around. So not everything at this time is going to be annotated, but we're working our way towards that. I wanted to bring your attention to the way we annotate antimicrobial resistance or genes in BV-BRC. There are two data sources. We go at the genome level and at the gene level. Now, at the genome level, we have been looking for large blocks of genomes that have been part of a larger study. And generally, these are put in the sequence read archive. We find them through publications. We go there. The authors who submitted it didn't want to necessarily put them in GenBank because there's so much data there and it would take a lot of their resources to do it. So they park it at the SRA, Sequence Read Archive. We go in and we grab that data. And we bring with it the metadata from the publications and biosample. And then we run machine learning algorithms on it to try to identify classifiers associated with resistance or susceptibility to various antibiotics. So outside of SRA and the publication, this is the only place you can get some of that data. At the gene level, we bring in information from publication, places like CARD, the NCBI Antimicrobial Resistance Database, which is also known as NARDO. We bring all that in. We have curators look at the data, and then they link those to functional roles or proteins that are used as a specialty database. So all of that is wrapped into RAS-TK. Every public genome, every private genome that goes through the system are blasted against these specialty databases. Now, at the genome level, we just have a certain number of species that we map classifiers to. But if you get those, you get a predicted antimicrobial resistant phenotype, and you can see predicted regions within the genome that are associated with antimicrobial resistance. At any other bacterial genome, every one of them gets annotated antimicrobial resistant genes, which you can see at the bottom of this graph. These are the genomes that we currently have or will soon be bringing into BV-BRC that will have those classifiers, meaning that we can predict a phenotype. The ones that are highlighted in red are the ones that we have right now. The ones that are in black are ones that we will be bringing into the resource early in 2025. So if you have one of these genomes, when you annotate it in BV-BRC, as long as you specify that it actually is Acinetobacter baumannii or one of these others, you should get a prediction of a phenotype of what it is resistant or susceptible to. So after you run the annotation pipeline, it's like Christmas morning. There are so many presents that you're about to get. This is what the landing page shows for the annotation job. It doesn't matter if you've annotated a bacterium, a phage, a virus, it all looks pretty much like this. Along the top, there are links to the genome landing page, the feature table, which are the genes, and the genome browser. At the bottom are all different forms of data that we think you might be interested in or that you've requested of us in the past. So what's annotated in red shows what those are, but the black part is the typical landing page. There's one that's different, and that's figure 4. Figure 4 gives you all of this, but it also gives you some other things that you'll have available with it. Annotation is really quick. This pipeline runs really fast, but it doesn't get integrated into the BV-BRC resource immediately. This takes a little time. We run the integration pipeline every hour on the hour. So let's say you submitted the job at 9:05 a.m. By 9:10, it might be done, depending upon how many people are using the service. And you will be able to see the job results, but you won't be able to see how it looks in the resource until the next hour. So that pipeline will run at 10 a.m. if you did it sometime at 9 a.m. And I'd start looking at like 9:05. If you click and try to look for it a little earlier, you'll get an error message that says, we can't find that genome. That's because it hasn't been integrated yet. But once it's there, it'll be available for only you to see, for you and anyone else you've shared your personal data with. So it'll be there, and you can see here that there is a phenotype associated with it if it is one of those specific species. So it's pretty cool. And all those tabs across the top are we're trying to bring in all the information. So you can see that annotation really gives you a lot of different tabs to look at, a lot of different information. So this is the landing page for the service. We're going to step through submitting one of these jobs, and we're going to use a data set from this particular study. And I think that you are really going to appreciate what this service provides in the course of these videos. So join me in the next one. I'll see you there.